Let's start by looking at culture methods, which are essential in microbiology for growing and studying microorganisms. These methods use various types of culture media to support microbial growth under controlled conditions. The different media come in many forms, like petri dishes, test tubes, and vials, with colors and compositions tailored to specific purposes. Understanding these basics sets the foundation for exploring how we cultivate and analyze microbes effectively. Now, what exactly is culture media? It's basically a nutrient-rich material prepared to help microorganisms grow in the lab. When we introduce microbes into this medium, they become what we call an inoculum. As these microbes grow and multiply in or on the medium, we refer to the resulting population as a culture. These definitions are key to grasping how microbiologists work with microbes in controlled environments. So, why do we use culture techniques? There are several important reasons. For example, isolating bacteria in pure cultures helps us study them in detail. Sometimes we need to grow enough bacteria to prepare antigens. We also test how bacteria respond to antibiotics, which is crucial for treatment decisions. Maintaining stock cultures ensures we have reference strains for future work. Additionally, cultures help demonstrate bacterial properties, type isolates for identification, and estimate viable bacterial counts. Each of these uses highlights the versatility of culture methods. Culture media can be classified in a few important ways. One way is by their special additives, things like enriched, selective, or differential media that serve specific purposes. Another classification is based on their physical state, solid, semi-solid, or liquid. Lastly, media can be grouped by their nutritional content, such as simple, complex, or synthetic media. This structured classification helps us choose the right media depending on what we want to achieve with our microbial cultures. Let's talk about simple media, also known as basal media. These are the most commonly used in routine lab work because they provide basic nutrients needed for many bacteria to grow. A good example is nutrient agar, which supports a wide range of bacterial species. Simple media are straightforward and effective for general cultivation, making them a staple in microbiology labs. Enriched media takes simple media a step further by adding substances like blood, serum, or egg to support the growth of fastidious organisms, those that require extra nutrients. For instance, blood agar and chocolate agar are popular enriched media that help grow bacteria that wouldn't thrive on simple media alone. Another example is LJ media, used for cultivating specific bacteria like Mycobacterium. These enriched media are crucial when dealing with demanding microbial species. Enrichment media are usually liquid and designed to promote the growth of certain organisms by providing enhanced nutrients. An example is BHI broth, which is enriched to support a broad range of bacterial growth. These media help increase the number of target organisms in a sample, making it easier to detect and study them. This selective promotion is very useful in microbiological investigations. Selective media contain additives that encourage the growth of specific bacteria while inhibiting others. This helps isolate particular organisms from mixed samples. Examples include DCA and TCBS media, which are tailored to support certain bacteria. By using selective media, we can focus on growing the microbes we're interested in, making identification and analysis more efficient. Differential media help us distinguish bacteria based on their characteristics, such as metabolic activities. For example, McConkey agar differentiates bacteria by producing colonies of different colors, depending on their ability to ferment lactose. This visual distinction is valuable because it allows us to identify and study bacteria more effectively without complex testing. Now, let's look at indicator and transport media. Indicator media show changes like color shifts caused by bacterial growth, which helps us identify specific reactions. Examples include McConkey agar, DCA, and TCBS media. Transport media, on the other hand, are designed to preserve delicate organisms during transport, especially when delays are expected. Stewart's media and APW are common examples. Both types serve important roles in maintaining and detecting bacteria under different conditions. Anaerobic media are specialized to support the growth of bacteria that can't tolerate oxygen. These media contain reducing substances that create an oxygen-free environment essential for cultivating anaerobes. Thioglycolate broth and RCM are typical examples. The growth patterns in these media help us identify and study anaerobic bacteria, which behave differently compared to those that thrive in oxygen. Culture methods can also be classified based on oxygen requirements of microorganisms. We have obligate aerobes that need oxygen, facultative anaerobes that can grow with or without oxygen, obligate anaerobes that cannot tolerate oxygen, microaerophilic organisms that require reduced oxygen levels, and capnophilic bacteria that need increased carbon dioxide. Understanding these categories helps us select appropriate culture conditions to support different microbes. There are several microbial culture techniques, each suited for different purposes. For example, streak culture isolates single colonies by spreading bacteria on agar. Stab culture involves inoculating deep into agar tubes. Lawn culture creates a uniform bacterial layer useful for antibiotic testing. Anaerobic methods cultivate bacteria without oxygen. Stroke culture is similar to streaking but done in tubes. Pour plate method mixes bacteria with molten agar to count colonies. Liquid medium culture grows bacteria in broth. Each technique offers unique advantages depending on the goal. Streak culture is a routine and effective way to isolate bacteria. It involves spreading the primary inoculum thinly across an agar plate in segments using a loop. The loop is sterilized between streaks to reduce bacterial density gradually. This method helps separate individual colonies from mixed samples, making it easier to study specific bacteria. To explain streak culture further, you start by streaking the inoculum in one area, then progressively dilute it across the plate in multiple segments. The loop is flamed and cooled between each section to prevent carrying too many bacteria forward. This results in isolated colonies in the final segments, which are essential for pure culture studies and identification. When we look at a streaked plate under the right conditions, we can see distinct colonies forming along the streak lines. The denser areas show more growth, while the later streaks reveal isolated colonies. This visual confirmation shows how effective streak culture is at separating bacteria for further analysis. Stab and stroke cultures are two related methods used in tubes. Stroke culture involves spreading bacteria on agar slopes, which provide a solid surface at an angle. Stab culture uses an inoculation needle to puncture the medium, allowing growth along the stab line. These methods help study bacterial growth patterns and characteristics in a controlled environment. Visually, slant tubes show agar solidified at an angle, providing a surface for stroke culture. Bacterial growth appears along this slant. Stab cultures show growth concentrated along the needle puncture line inside the medium. These distinct growth patterns help differentiate bacterial behaviors and test their properties effectively. Stab cultures show a clear line of growth where the inoculation needle penetrated the medium. The pour plate method is a quantitative technique where melted agar is cooled, mixed with diluted inoculum, and poured into sterile petri dishes. After incubation, colonies grow within the agar and on the surface. We count these colonies to estimate bacterial numbers. This method is important for quantifying bacteria in samples accurately. With serial dilution in the pour plate method, the number of colonies decreases as dilution increases. Plates with too many colonies become confluent and uncountable, while plates with fewer colonies allow precise counting. This trend helps us find the right dilution for accurate bacterial quantification. Lawn culture produces a uniform bacterial growth across the agar surface. It's commonly used for antibiotic sensitivity testing. By applying antibiotic discs on the lawn, the bacteria grow evenly, allowing clear zones of inhibition to form around effective antibiotics. This practical approach helps guide treatment choices by showing which antibiotics are effective against the bacteria.
Anaerobic cultivation methods are designed to isolate bacteria that require oxygen-free environments. We create anaerobiosis using techniques like Macintosh and Field's jar, anaerobic gas pack method, or evacuation replacement. These methods, combined with anaerobic media containing reducing agents, allow us to grow and study anaerobic bacteria that wouldn't survive otherwise. The anaerobic gas pack method is a convenient single-use technique. It uses chemical pellets that generate hydrogen and carbon dioxide when water is added. Inoculated plates are placed inside a jar with the gas pack and sealed tightly. This setup creates an anaerobic atmosphere ideal for growing oxygen-sensitive bacteria.
A Macintosh and File ice jar is a specialized container for anaerobic cultivation. It includes a lid with a gasket, clamps, palladium catalyst pellets, and chemicals that produce hydrogen and carbon dioxide gases. An indicator shows when anaerobic conditions are achieved. Plates inside the jar grow bacteria in an oxygen-free environment, enabling study of anaerobes. The candle jar method creates a microaerophilic environment by placing a lit candle inside a sealed jar with bacterial cultures. As the candle burns, it consumes oxygen, lowering its concentration and increasing carbon dioxide levels. This controlled atmosphere supports the growth of bacteria that need reduced oxygen, like microaerophiles and capnophilic organisms, making it a simple yet effective cultivation technique.